Evolution tried out creatures so strange that when their fossils were first discovered, scientists refused to believe they could be real. But the most bizarre experiments didn't have teeth, didn't have eyes, and didn't even move. They were plants, not the harmless green backdrop we see today, but towering biological weapons that poisoned the air, choked out entire landscapes, and developed hunting strategies that would make modern predators jealous. Before forests became peaceful sanctuaries, they were battlegrounds where plants didn't just compete for sunlight, they actively killed, trapped, and consumed anything that wandered too close. These botanical monsters evolved defenses so extreme and appetites so ruthless that animals had no choice but to adapt or die. The ancient world was ruled by vegetation that makes today's most dangerous plants look like garden flowers. If you've ever thought plants were the passive victims of evolution's story, get ready for a truth that rewrites everything we know about life on Earth. The Proto-Taxites Towers. Standing in a Silurian landscape 400 million years ago would have felt like stepping onto an alien planet designed by a fever dream. The horizon stretched flat and barren, interrupted only by massive biological pillars rising from the ground like organic skyscrapers. These weren't trees. Trees hadn't been invented yet. These were proto-taxites, and they dominated the landscape in a way nothing else could. Each one stood up to 8 meters tall, roughly the height of a 3-story building. But their trunks were only a meter wide. They looked like giant mushrooms stretched vertically, or chimneys made of living tissue. For over a century after their discovery in 1859, scientists couldn't figure out what they were looking at. The confusion was understandable. Proto-taxites didn't fit any category. Early paleobotanists thought they might be conifers, some bizarre early tree that evolved a strange growth pattern. But the internal structure was all wrong. Cross-sections showed a network of interwoven tubes that didn't match any known wood. The tubes were too regular, too organised, and too uniform. Some researchers proposed they were giant algae that somehow grew on land, but that theory collapsed when chemical analysis showed the tissues weren't photosynthetic. Others suggested they were lichens, a symbiotic relationship between fungi and algae, but the scale made no sense. Lichens don't grow 8 meters tall. The breakthrough came from analysing the tube structure at a microscopic level. The tubes were hyphae, the thread-like structures that make up fungal bodies. Proto-taxites wasn't a plant at all. It was a fungus, the largest fungus that has ever existed on land. But even that classification created new mysteries. Modern fungi decompose dead material or form partnerships with plant roots. They don't grow into towers. They don't dominate landscapes. They certainly don't stand alone as the tallest organisms in their ecosystem. Proto-taxites broke every rule about what fungi were supposed to do. The world Proto-taxites inhabited had no competition for vertical space. The tallest plants at the time were primitive mosses and early vascular plants like Cooksonia, which barely reached ankle height. In this low-lying landscape, Proto-taxites towered like biological monuments. Their height wasn't just impressive, it was functional. Being tall meant their spores could catch wind currents and spread across vast distances. While other organisms crawled along the ground, Proto-taxites released reproductive cells from 8 meters up, giving them a dispersal advantage nothing else possessed. But what did these fungal towers eat? Modern fungi feed on dead organic matter, breaking down fallen trees, dead animals, and decomposing leaves. Proto-taxites lived in a world where none of those things existed in abundance. There were no forests to create leaf litter, no large animals to leave carcasses, and very little biomass accumulating on the ground. Some scientists proposed that Proto-taxites formed mutualistic relationships with early plants, exchanging nutrients in a primitive version of the mycorrhizal networks that help modern forests communicate. Others suggest it fed on microbial mats or primitive algae that covered wet soil. A more unsettling theory suggests prototaxites may have been parasitic. Evidence from fossil sites shows that some specimens grew in clusters, their bases intertwined with early plant material. Chemical signatures in the preserved tissue hint at nitrogen levels too high for a purely decomposer lifestyle. If prototaxites parasitized living plants, it would explain how it sustained such enormous size in a nutrient-poor environment. It would also make it one of the earliest examples of a plant-killing organism, a fungal predator that drained its hosts while standing above them like a monument to their demise. The strangest detail about prototaxites is how it disappeared. For nearly 50 million years, these towers defined terrestrial landscapes. Then, as forests began to rise and true trees evolved, prototaxites vanished from the fossil record. The timing suggests competition. Once trees grew tall enough to shade the ground and create their own fungal partnerships, prototaxites lost their advantage. Its height, once unmatched, became irrelevant. Its feeding strategy, once unique, was outcompeted by fungi that specialized in breaking down wood. Evolution moved on, and prototaxites became a relic of a stranger, simpler world. The lepidodendron forests. Walking through a Carboniferous forest 300 million years ago would have been like wandering through a nightmare designed by nature's most reckless architect. Trees stretched 40 meters into the air, their trunks covered in diamond-shaped scars that made them look like they were armored in reptilian scales. These weren't the sturdy oaks or flexible pines we know today. These were lepidodendron, and they were built on a biological gamble so extreme that it eventually collapsed their entire ecosystem. Every aspect of their anatomy was a compromise between growth and survival, and when the climate shifted, that compromise killed them all. Lepidodendron belonged to a group called lycopsides, relatives of modern club mosses. But while today's club mosses grow a few centimeters tall and hide in the understory, lepidodendron evolved into giants. Their trunks could reach over a meter in diameter, and their root systems spread across the forest floor in shallow, tangled networks called stigmaria. These roots weren't deep. Lepidodendron had no taproot to anchor it firmly into the ground. Instead, it relied on sheer width, spreading its roots horizontally to cover as much surface area as possible. This strategy worked in the waterlogged swamps of the Carboniferous, where the ground was soft and saturated. But it also made lepidodendron vulnerable. A strong storm could topple entire groves. The bark was the most distinctive feature. Those diamond-shaped scars weren't decorative, they were leaf bases. Lepidodendron didn't shed its leaves the way modern trees do. Instead, as the tree grew, old leaves fell off and left behind permanent marks on the trunk. The scars formed a geometric pattern so precise that early paleobotanists thought they were looking at fossilized snakeskin. Each scar represented a point where the tree had once photosynthesized, and as the tree aged, these marks became a historical record of its growth. The pattern was beautiful, but it was also a sign of inefficiency. Lepidodendron kept adding height without adding strength. The internal structure of lepidodendron was even stranger. Most trees build thick, solid wood to support their weight. Lepidodendron didn't. Its trunk was mostly soft tissue supported by a thin outer shell of rigid material. The center was filled with a spongy pitch that provided almost no structural support. This design was light, allowing the tree to grow tall quickly without investing energy in dense wood. But it also meant lepidodendron trunks were fragile. Fossil sites show evidence of mass die-offs where entire forests collapsed, their trunks snapped at the base, or toppled sideways into the swamp. The reproductive strategy of lepidodendron was just as extreme. Instead of producing seeds gradually over many years, lepidodendron saved all its energy for one massive reproductive event. When the tree reached full height, it would produce enormous cones, some over half a metre long, packed with millions of spores. This single reproductive burst drained the tree completely. After releasing its spores, Lepidodendron died. It was a biological all-in bet. Grow fast, grow tall, reproduce once, and then collapse. In stable environments, this strategy worked. Lepidodendron forests dominated the Carboniferous for millions of years, covering vast stretches of what is now Europe and North America. But stability didn't last. The Carboniferous climate was warm, wet, and consistent. Perfect conditions for Lepidodendron's shallow roots and water-dependent reproduction. Then the climate began to dry. Swamps drained, water tables dropped, and seasonal droughts became common. Lepidodendron couldn't adapt. Its roots were too shallow to reach deeper water. Its reproductive cycle required constant moisture. Its fragile trunks couldn't withstand the stronger winds that came with drier climates. Within a few million years, Lepidodendron forests collapsed. The giants that had dominated the landscape for 50 million years went extinct, replaced by seed plants and conifers that could survive in drier conditions. The fossil record preserves Lepidodendron in extraordinary detail. Entire fossilized forests exist where the trees fell and were buried in swamp sediment before they could decay. These sites, known as coal forests, show the strange ecology Lepidodendron created. The forest floor was a tangle of shallow roots, fallen trunks, and standing dead trees. Oxygen levels in the Carboniferous were much higher than today, and fires were common. Evidence suggests that Lepidodendron forests burned frequently, the flames spreading through the dry, spongy wood and consuming entire groves in hours. The trees would regrow from spores, only to burn again in an endless cycle of destruction and regeneration. The legacy of Lepidodendron is buried beneath our feet. When these forests died and collapsed into the swamps, their soft tissue didn't decay. It turned into coal. The massive coal deposits mined today in Europe, North America, and Asia are the compressed remains of Lepidodendron forests. Every piece of coal is a fragment of a tree that gambled everything on height and lost when the world changed. These weren't just plants. They were architects of the carbon cycle, locking away so much atmospheric carbon that they may have triggered an ice age. Their rise reshaped the atmosphere. Their extinction reshaped geology. And their fossils still fuel the modern world. The Sigillaria swamps. If Lepidodendron was the reckless giant of the Carboniferous, Sigillaria was its more cautious but equally strange cousin. These trees stood just as tall, sometimes reaching 35 metres, but their approach to survival was entirely different. Where Lepidodendron spread wide and grew fast, Sigillaria grew narrow and invested in a stronger root system. Its trunk was covered in vertical rows of oval scars, giving it the appearance of being wrapped in braided rope. These scars marked where leaves had once attached, but unlike Lepidodendron's chaotic diamond pattern, Sigillaria's scars formed neat, orderly columns. It looked like a tree designed by an engineer obsessed with symmetry. Sigillaria's roots were its greatest innovation. While Lepidodendron relied on shallow stigmaria that spread horizontally, Sigillaria developed a more advanced root structure called a rooting organ. This structure anchored the tree more securely, allowing it to grow in slightly drier environments than Lepidodendron could tolerate. Fossil evidence shows Sigillaria often grew at the edges of swamps, in transitional zones where water levels fluctuated seasonally. This adaptability gave it a survival advantage. When droughts hit, Lepidodendron forests died. Sigillaria forests held on. But Sigillaria wasn't invincible. Its trunk structure was still fundamentally weak. Like Lepidodendron, it relied on a thin outer shell and a soft, spongy interior. Cross-sections of fossilized Sigillaria trunks reveal the same fragile design. Rigid bark surrounding tissue that provided almost no support. The tree compensated by growing narrower, reducing the stress on its trunk, but this created new problems. A narrow trunk meant less surface area for photosynthesis. Sigillaria had to produce more leaves to gather the same amount of sunlight, and those leaves needed constant water. In dry conditions, Sigillaria couldn't sustain itself. The reproductive strategy of Sigillaria was equally demanding. Like Lepidodendron, it reproduced using spores rather than seeds. But Sigillaria's cones were smaller and produced less frequently. Instead of one massive reproductive event, Sigillaria spread reproduction across several years. This strategy was more sustainable, allowing the tree to survive longer and produce multiple generations of spores. But it also meant Sigillaria reproduced more slowly. In rapidly changing environments, slow reproduction was a death sentence. By the time Sigillaria could produce enough spores to adapt to new conditions, the environment had already shifted again. One of the strangest aspects of Sigillaria is how it interacted with other plants. Fossil sites show that Sigillaria often grew in dense stands where the trees were packed so closely together that their trunks nearly touched. This tight spacing created a shaded understory where almost nothing else could grow. Sunlight couldn't penetrate the canopy, and the shallow root systems of Sigillaria absorbed most of the available water. The forest floor beneath Sigillaria groves was a biological dead zone, bare soil punctuated by fallen trunks and rotting vegetation. Sigillaria didn't just dominate its environment, it suffocated it. This dominance came with consequences. Sigillaria forests altered local water cycles. Their shallow roots drew moisture from the soil faster than rain could replenish it. Over time, this caused localized droughts that killed other plant species and prevented new vegetation from establishing. The result was an ecosystem locked in stasis, Sigillaria forests that couldn't evolve and couldn't be replaced. When the climate finally shifted and swamps dried permanently, these forests had no successors. The land Sigillaria once occupied turned into barren plains. The fossil record shows that Sigillaria survived longer than Lepidodendron, clinging to wet environments even as the climate dried. But survival wasn't the same as thriving. Late Carboniferous fossils show Sigillaria trees growing smaller, their trunks thinner and their cones fewer. The species was in decline, struggling to maintain itself in a world that no longer suited it. By the early Permian, Sigillaria had vanished. Its extinction marked the end of the giant lycopside forests and the beginning of a new era dominated by seed plants. The legacy of Sigillaria is preserved in coal seams alongside Lepidodendron. Miners in the 19th century often found fossilized Sigillaria trunks standing upright in coal deposits, preserved exactly as they had fallen 300 million years ago. These fossil forests provided the first clear evidence that coal was biological in origin, formed from compressed plant material rather than mineral deposits. The discovery revolutionized geology and gave scientists their first glimpse into the strange, swampy world of the Carboniferous. The Archaeopteris Revolution. Before flowering plants, before conifers, before even true seeds, there was Archaeopteris, a tree that looked almost modern but operated on principles evolution would later abandon. It stood 30 meters tall with a trunk that could reach a meter in diameter, and it had branches covered in fern-like fronds that turned the forest floor into a mosaic of shadow and light. From a distance, Archaeopteris forests looked like they belonged in the present day. Up close, the illusion shattered. These trees reproduced using spores, not seeds. Their wood was primitive, lacking the growth rings and complex vascular systems that define modern trees. And their ecological impact was so profound that they literally reshaped the planet's surface. Archaeopteris evolved during the late Devonian, around 370 million years ago. At the time, land plants were still relatively small. The tallest organisms were early lycopsids and primitive ferns that barely reached a few meters in height. Archaeopteris broke that ceiling. Its height gave it access to sunlight no other plant could reach, and its extensive root system allowed it to anchor itself firmly in the ground. For the first time in Earth's history, forests had depth. There was a canopy, an understory, and a forest floor. 3 distinct ecological zones where different organisms could evolve specialized lifestyles. The roots were the real revolution. Before Archaeopteris, plant roots were shallow and simple, designed mainly for absorbing water. Archaeopteris developed deep, branching roots that penetrated soil to depths of several meters. These roots didn't just anchor the tree, they broke apart rock. As the roots expanded, they cracked bedrock, creating fractures that filled with organic material and water. Over thousands of years, this process transformed barren rock into fertile soil. Archaeopteris forests created the conditions necessary for other plants to evolve. Without Archaeopteris, the rich soils that support modern ecosystems might never have developed. But deep roots had another effect, one that changed the planet's climate. When roots break down rock, they release minerals that react with carbon dioxide in the atmosphere. This chemical weathering removes CO2 from the air and locks it into soil and sediment. Archaeopteris forests covered vast areas of the late Devonian world, and their roots pulled so much carbon dioxide out of the atmosphere that global temperatures dropped. Some scientists believe Archaeopteris forests triggered one of the ice ages that defined the late Devonian, cooling the planet and reshaping ocean currents. The reproductive biology of Archaeopteris was caught between 2 evolutionary stages. It produced spores like ferns and mosses, releasing them from structures that looked like primitive cones. But unlike earlier spore-producing plants, Archaeopteris showed signs of developing separate male and female reproductive structures. Some fossils preserve small spores that likely developed into male gametophytes, while others show larger spores that became female gametophytes. This separation of sexes was a crucial step toward the evolution of seeds, which would emerge in later plant lineages. Archaeopteris didn't develop true seeds, but it came close enough that paleobotanists consider it a transitional form. The wood of Archaeopteris reveals another evolutionary experiment. Modern trees produce secondary wood, adding new layers each year to create growth rings. Archaeopteris produced secondary wood, but its growth pattern was irregular. Some years it added thick layers, other years it added almost nothing. The wood lacked the complex vessel systems that transport water efficiently in modern trees. Instead, Archaeopteris relied on simpler tracheids, elongated cells that moved water slowly through the trunk. This inefficiency limited how tall Archaeopteris could grow and how much water it could transport, but it was still far more advanced than anything that came before. One of the most fascinating discoveries about Archaeopteris came from studying its fossilized leaves. The fronds looked like fern leaves, with small leaflets arranged along a central stem. But microscopic analysis revealed stomata, tiny pores that control gas exchange. The density and arrangement of these stomata suggest Archaeopteris was remarkably efficient at photosynthesis. It could capture and process sunlight more effectively than earlier plants, giving it a competitive advantage in shaded environments. This efficiency allowed Archaeopteris to grow in dense stands where other plants would have struggled. The extinction of Archaeopteris coincided with one of the most devastating events in Earth's history, the late Devonian mass extinction. This extinction wiped out 75% of all species, and Archaeopteris was among the casualties. The exact cause remains debated, but evidence points to a combination of climate change, ocean anoxia, and possibly an asteroid impact. Whatever the trigger, Archaeopteris forests vanished. The ecological niche they created didn't disappear though. Seed plants evolved to fill the gap, taking the lessons Archaeopteris had demonstrated: deep roots, tall trunks, efficient photosynthesis, and refining them into forms that could survive the changing world. the calamites' jungles. Imagine a forest where every tree looked like a gigantic version of a horsetail weed, with segmented trunks divided into hollow sections like bamboo, branches sprouting in perfect whorls, and leaves reduced to tiny scales. That was the world of calamites, and it dominated wetland environments throughout the Carboniferous. These weren't small plants. Calamites could grow 20 metres tall, with trunks 30 centimetres in diameter. They turned swamps into dense jungles where navigation was nearly impossible, and where the architecture of the forest followed geometric rules that seemed almost artificial. Calamites belong to the sphenopsids, the same group that includes modern horsetails. But while today's horsetails grow knee-high in ditches and wetlands, calamites evolved into giants. The key to their size was their hollow trunk structure. Unlike solid trunk trees, calamites' trunks were divided into segments separated by nodes, similar to bamboo. Each segment was hollow, reducing the tree's weight and allowing it to grow tall without investing massive amounts of energy in solid wood. This design was efficient, but it was also fragile. Fossil evidence shows that calamites' trunks often broke at the nodes, especially during storms or when the tree grew too tall. The root system of calamites was equally unusual. Instead of a single taproot or a spreading network of lateral roots, calamites produced horizontal underground stems called rhizomes. These rhizomes spread through the soft swamp mud, sending up new shoots at regular intervals. A single calamite's individual could produce dozens of vertical trunks, creating what looked like a grove, but was actually one interconnected organism. This clonal growth strategy allowed calamites to colonize wetlands rapidly, spreading across open water and turning shallow lakes into dense thickets. The branches of calamites grew in whorls, circular arrangements where multiple branches emerged from a single node. Each whorl contained between 6 and 12 branches, depending on the species, and each branch subdivided into smaller branches following the same pattern. The result was a fractal-like structure that repeated at every scale. From a distance, calamites' forests looked almost mechanical, as if they had been assembled rather than grown. This geometric precision wasn't just aesthetic, it was functional. The whorled branch pattern maximized surface area for photosynthesis while minimizing the tree's weight. The leaves of calamites were tiny, barely more than scales fused to the branches. They contributed almost nothing to photosynthesis. Instead, the branches themselves were green and photosynthetic, functioning more like the stems of modern horsetails. This design made calamites remarkably drought-resistant for a wetland plant. When water levels dropped, the tree could shed its branches and survive on photosynthesis from its green trunk alone. When water returned, new branches would sprout from the nodes and the tree would resume normal growth. Reproduction in calamites followed a pattern similar to modern horsetails, but on a vastly larger scale. The trees produced cones at the tips of their branches, each cone containing thousands of spores. These cones were arranged in clusters, and when mature, they released spores in massive clouds that could travel kilometers on the wind. Fossil sites preserve layers of calamite spores so thick that they form distinct strata in the rock, evidence of reproductive events that blanketed entire regions. One of the strangest aspects of calamite's ecology was its relationship with fire. The Carboniferous atmosphere contained 35% oxygen, much higher than the 21% we breathe today. This high oxygen level made fires more frequent and more intense. Calamites' forests burned regularly, and fossil charcoal shows evidence of repeated fire events. But calamites didn't just tolerate fire, it thrived on it. The rhizome system allowed the plant to re-sprout quickly after burns, and the lack of competitors in recently burned areas gave calamites an advantage. Fire became part of the life cycle, clearing away dead material and opening space for new growth. The fossil record preserves calamites in exceptional detail. In some coal deposits, entire fossilized calamites forests remain standing, their hollow trunks filled with sediment but still upright. These sites provide snapshots of Carboniferous wetlands frozen in time, showing how densely packed Calamites forests could be. In some areas, trunks stood less than a metre apart, creating an almost impassable tangle. Animals that lived in these environments had to navigate by climbing or by following the rare open channels between rhizome clumps. The extinction of Calamites followed the same pattern as other Carboniferous giants: climate change and the drying of swamps. As the Permian period began and wetlands shrank, Calamites lost its habitat. The rhizome strategy that had been so successful in swamps became a liability in drier environments. The shallow underground stems couldn't reach deep water, and the hollow trunks couldn't survive seasonal droughts. Within a few million years, Calamites dwindled from dominant forestry to marginal wetland plant. By the Triassic, the giant sphenopsids were gone, replaced by their tiny descendants that still grow in ditches today. The Cordaites' canopies. While Lycopsids and Sphenopsids dominated the swamps, a different lineage was quietly evolving in slightly drier environments: the Cordaites. These trees looked deceptively modern, with straight trunks, simple branch patterns and long strap-like leaves. They could grow 40 metres tall, rivaling the largest lepidodendron, but their internal structure was far more advanced. Chordates were among the first plants to develop true wood with growth rings, efficient water transport systems, and reproductive structures that foreshadowed the evolution of conifers. They represented evolution's first serious attempt at building a tree that could survive outside of swamps. The leaves of chordates were its most distinctive feature. Unlike the small, scale-like leaves of Lycopsids or the branching fronds of ferns, chordates' leaves were long, narrow blades that could reach over a metre in length. They grew in spiral clusters at the ends of branches, creating dense canopies that blocked sunlight from reaching the forest floor. Fossil impressions show that these leaves had parallel veins running their entire length, a design that allowed efficient water transport and made the leaves surprisingly resilient to damage. Even when torn or partially eaten, chordates' leaves continued to photosynthesize. The wood of chordates marked a major evolutionary leap. Earlier trees like lepidodendron and calamites had soft, spongy tissue that provided minimal support. Chordates developed dense secondary wood composed of tightly packed cells called tracheids. These cells were elongated and overlapping, creating a strong, rigid structure that could support significant weight. Cross-sections of fossilized chordates' trunks show clear growth rings, indicating that the trees experience seasonal growth cycles. This seasonal growth suggests chordates lived in environments with variable climates, areas where water availability changed throughout the year. The root system of chordates was deep and branching, far more sophisticated than the shallow stigmaria of Lycopsids. Fossil root systems show chordates' roots penetrating several metres into the soil, anchoring the tree firmly and allowing it to access groundwater during dry periods. This deep root system gave Chordaites a crucial advantage. It could grow in upland environments where other Carboniferous trees couldn't survive. While Lepidodendron and Sigillaria were confined to swamps, Chordaites colonized hillsides, riverbanks, and well-drained lowlands. Reproduction in chordates represented another evolutionary innovation. Instead of producing spores in simple cones like Lycopsids, chordates developed separate male and female cones with more complex structures. The male cones produced pollen, which the wind carried to female cones. The female cones contained ovules, structures that would eventually evolve into seeds. Chordate ovules were large and protected by specialized scales, a design that foreshadowed the seed cones of modern conifers. This reproductive strategy was more efficient than spore-based reproduction, giving chordate seedlings a better chance of survival in competitive environments. One of the most fascinating aspects of chordate's ecology was its relationship with early insects. The Carboniferous saw the evolution of giant dragonflies, massive millipedes, and the first plant-eating insects. Fossil evidence shows that insects fed on chordates leaves, creating characteristic damage patterns preserved in the fossil record. Some leaves show bite marks along the edges, while others have holes punched through the blade. This suggests chordates was a primary food source for herbivorous insects, making it a keystone species in Carboniferous forest ecosystems. The distribution of chordates was remarkably wide. Fossils have been found on every continent, including Antarctica, indicating that chordates forests covered vast areas of the late Carboniferous and early Permian world. These forests provided habitat for early reptiles, amphibians, and insects, creating complex food webs that resembled modern forest ecosystems. Some paleontologists argue that chordates forests were the first true forest ecosystems, environments where multiple layers of vegetation supported diverse communities of animals. The extinction of chordates was gradual rather than sudden. As conifers evolved and diversified during the Permian, they began to outcompete chordates in many environments. Conifers had more efficient reproductive systems, more drought-resistant leaves, and better adaptations for surviving in seasonal climates. Chordates persisted into the early Triassic in some regions, but by the Middle Triassic they had vanished. Their descendants, the conifers, inherited the ecological niches chordates had pioneered and went on to dominate forests for the next 200 million years. The Glossopteris monopoly. In the southern hemisphere during the Permian, one plant genus achieved something no other plant before or since has managed. It became the only significant tree across an entire supercontinent. Glossopteris dominated Gondwana so completely that fossil sites from this period contain almost nothing else. Forests stretched across Antarctica, Australia, India, South Africa, and South America, and every tree in those forests was Glossopteris. This wasn't just dominance, it was a botanical monopoly that reshaped the southern hemisphere's ecology and left a fossil record so distinctive that it became key evidence for continental drift. Glossopteris trees weren't giants. Most species grew between 5 and 10 metres tall, with trunks rarely exceeding 30 centimetres in diameter. But what they lacked in size, they made up for in numbers and adaptability. Glossopteris could grow in conditions that killed other plants, cold climates, poor soils, and environments with long periods of darkness. The Permian southern hemisphere experienced seasonal extremes, with some regions spending months in twilight or complete darkness during winter. Glossopteris evolved to handle this by developing deciduous habits, shedding its leaves during dark months and regrowing them when sunlight returned. The leaves were Glossopteris's signature feature. They were tongue-shaped, broad, and covered with a network of veins that formed a distinctive mesh pattern. This reticulate venation was unique. No other Permian plant had anything like it. The veins created a strong internal framework that supported large leaf blades while maintaining flexibility. Fossil leaves show remarkable preservation, with vein patterns visible in such detail that individual species can be identified based on leaf structure alone. These leaves weren't just functional, they were evolutionary experiments in vein architecture that wouldn't appear again until flowering plants evolved over 100 million years later. Reproduction in Glossopteris followed a pattern that puzzled paleobotanists for decades. The trees produced seeds, but the seeds weren't contained in typical cones. Instead, they grew on specialized leaf-like structures called fructifications. These organs looked like leaves that had been modified to hold seeds, with the seeds attached directly to the surface, rather than enclosed in protective scales. This reproductive structure was so unusual that some scientists initially classified the seed-bearing organs as separate plants. Only after finding them consistently associated with Glossopteris leaves did researchers realize they belonged to the same organism. The wood of Glossopteris shows clear growth rings, indicating that the trees experience strong seasonal growth cycles. Ring patterns reveal that Glossopteris forests grew rapidly during the short summer growing season and entered dormancy during long, dark winters. Some fossil trunks show over 100 growth rings, suggesting individual trees could live for over a century. This longevity allowed Glossopteris to dominate landscapes for millions of years, with individual trees witnessing generations of animals come and go beneath their canopies. One of the most remarkable discoveries about Glossopteris came from studying its root systems. Fossil sites in Antarctica preserve Glossopteris roots penetrating permafrost soils, evidence that these trees could survive in conditions cold enough to freeze the ground seasonally. The roots show adaptations for extracting water from frozen soil, including specialized tissue that prevents ice crystal damage. This cold tolerance explains how Glossopteris could dominate polar regions of Gondwana, growing in latitudes where 6 months of darkness made photosynthesis impossible for half the year. The ecological impact of Glossopteris was profound. By monopolizing forest ecosystems across Gondwana, it created uniform habitats that stretched across thousands of kilometers. Animals that evolved to feed on Glossopteris could spread across the entire supercontinent without encountering significant ecological barriers. This uniformity may have contributed to the rapid diversification of herbivorous reptiles during the Permian, as plant eaters developed specialized adaptations for processing Glossopteris leaves, wood and seeds. The fossil record preserves evidence of a complex relationship between Glossopteris and insects. Fossil leaves frequently show damage patterns indicating herbivory, holes, bite marks and galleries where insects burrowed through leaf tissue. Some leaves show evidence of galling, where insects induced the plant to produce abnormal growth that provided food and shelter. These interactions suggest Glossopteris forests supported diverse insect communities, which in turn provided food for predatory insects, early spiders, and small vertebrates. The extinction of Glossopteris came at the end of the Permian during the worst mass extinction in Earth's history. The Permian Triassic extinction event killed over 90% of all species, and Glossopteris was among the casualties. The exact cause of its extinction remains debated, but the combination of volcanic eruptions, climate change, and ocean acidification created conditions that even adaptable Glossopteris couldn't survive. Within a few million years, the forests that had dominated Gondwana for 50 million years were gone, replaced by entirely new plant communities that had no connection to Glossopteris lineages. The Medulosa seed ferns. Evolution tried to make seeds work in plants before it figured out how to do it properly, and Medulosa was one of the strangest results of that experiment. These weren't true ferns despite their fern-like leaves, and they weren't quite trees despite their woody trunks. They were seed ferns, plants that combined the appearance of ferns with the reproductive biology of seed plants. Medulosa could grow up to 8 meters tall, with trunks covered in a tangled mass of aerial roots that made them look like they were standing on stilts. The whole structure seemed barely stable, as if evolution had sketched out a rough draft and forgotten to refine it. The trunk of Medulosa was a biological mess, a thick core of soft tissue surrounded by a chaotic arrangement of vascular bundles that had no clear organization. Most plants organize their vascular tissue in neat rings or patterns that allow efficient water transport. Medulosa scattered its vascular bundles randomly throughout the trunk, creating a structure that looked inefficient and poorly designed. But this chaos served a purpose. The multiple vascular bundles provided redundancy. If one pathway was damaged by insects or disease, others could compensate. This made Medulosa remarkably resilient to partial damage. The aerial roots were Medulosa's most distinctive feature. These roots didn't grow underground, but instead emerged from the trunk several meters above the ground and descended in tangled clusters. The roots formed a dense network that surrounded the base of the trunk like a cage, providing structural support and protecting the vulnerable soft tissue at ground level. Some fossil specimens preserve these root systems intact, showing that individual roots could grow several centimetres thick and weave together into structures strong enough to support the entire tree. The leaves of Medulosa were elaborate, fern-like fronds that could reach 2 metres in length. Each frond is divided and subdivided into hundreds of small leaflets, creating a massive surface area for photosynthesis. These fronds grew from the top of the trunk in a crown, giving Medulosa a palm-like appearance. But unlike palm leaves, Medulosa fronds were deciduous in some species, shedding during unfavourable seasons and regrowing when conditions improved. Fossil sites preserved leaf beds where thousands of shed Medulosa fronds accumulated, creating distinctive layers in the rock. The seeds of Medulosa were enormous. Some species produce seeds over 10 centimetres long, among the largest seeds ever documented in the plant kingdom. These seeds weren't protected by cones or fruits, but grew exposed on specialised branches. Each seed was covered in a thick, fleshy layer that probably attracted animals who ate the flesh and dispersed the seeds in their droppings. This animal dispersal strategy was rare in Permian plants and suggests Medulosa had co-evolved with seed-eating reptiles or early mammals. One of the strangest discoveries about Medulosa reproduction came from studying the pollen-producing structures. Unlike most seed plants that produce pollen in simple cones, Medulosa developed elaborate organs covered in specialized scales and tentacle-like projections. These structures looked more like sea anemones than plant parts, and their function remained mysterious until microscopic analysis revealed they produce pollen with a sticky coating. This coating helped the pollen adhere to the female reproductive structures, improving fertilization success in environments where wind pollination was unreliable. The ecological role of Medulosa was that of an understory tree in Carboniferous and Permian forests. While giants like Lepidodendron and Chordates dominated the canopy, Medulosa thrived in the shaded middle layer where its large fronds could capture filtered sunlight. Fossil sites show Medulosa grew in mixed forests alongside Lycopsids, Chordates and early conifers, creating complex layered ecosystems. The aerial root systems may have provided habitat for climbing animals, insects and small vertebrates that nested in the tangled root masses. Evidence from fossil sites suggests Medulosa was remarkably widespread, with species found in North America, Europe and Asia. Different species adapted to different environments. Some grew in wet lowlands, while others colonized drier upland areas. This adaptability allowed Medulosa to persist through the climate changes of the late Carboniferous and early Permian. But like many Permian plants, Medulosa couldn't survive the end Permian extinction. The lineage vanished completely, leaving no descendants. The ecological niche it occupied, mid-canopy seed fern, wasn't refilled until flowering plants evolved similar growth forms over 100 million years later. The Neuropterus canopy. In the shadow of the Carboniferous giants grew Neuropterus, a seed fern with leaves so delicate and beautifully arranged that early paleobotanists refused to believe they belonged to the same harsh, swampy world as Lepidodendron and Calamites. These weren't towering trees, but smaller plants, rarely exceeding 5 metres in height, with compound leaves that spread in elegant fans. Each leaf was divided into dozens of rounded leaflets, arranged along a central stem, creating a lace-like pattern that seemed too fragile for the brutal ecosystems of the Carboniferous. But appearances were deceiving. Neuropterus was a survivor that outlasted many of its larger neighbours. The leaflets of Neuropterus were its defining characteristic. Each individual leaflet was small, rounded, and attached to the main stem by a narrow stalk. The arrangement created a feather-like appearance, with leaflets overlapping slightly to maximise light capture while minimising shading between individual segments. Fossil preservation of these leaves is often exceptional, with cellular detail visible under magnification. Scientists can see the stomata, the vein patterns, and even evidence of insect damage preserved in stunning clarity. What made Neuropteris successful wasn't size but flexibility. While other carboniferous plants committed to specific environments, Lepidodendron to waterlogged swamps, Chordates to well-drained uplands, Neuropteris adapted to intermediate conditions. It grew in areas where water levels fluctuated seasonally, where soil conditions varied, and where competition from larger plants created unstable light conditions. This adaptability came from its growth strategy. Neuropteris could grow rapidly when conditions were favourable and enter dormancy when stressed, shedding its delicate leaves and surviving on stored reserves in its woody stem. The reproductive structures of Neuropteris were only recently understood. For over a century, paleobotanists found the leaves but couldn't locate the seeds. The mystery was solved when detailed excavations revealed that Neuropteris seeds grew on entirely different-looking structures that had been classified as separate plants. These seed-bearing organs were fleshy, complicated, and bore no resemblance to the delicate foliage. Once researchers connected the organs to the leaves through consistent association in fossil beds, the picture became clear. Neuropteris had evolved a strategy where vegetative and reproductive parts looked nothing alike. The seeds themselves were sophisticated. They had multiple protective layers, internal tissue that nourished the developing embryo, and surface structures that may have helped with water absorption or animal dispersal. Size-wise, they fell between the giants of Medulosa and the smaller seeds of other seed ferns, suggesting a middle ground reproductive strategy. The seeds were numerous enough to ensure some survived, but large enough that each had significant energy reserves for germination. Evidence from coal deposits shows that Neuropteris was extraordinarily abundant. In some fossil localities, Neuropteris leaves make up the majority of plant fossils, suggesting these plants grew in dense thickets that covered large areas. This abundance made Neuropteris a crucial component of carboniferous ecosystems. Its leaves provided food for insects and early herbivorous reptiles. Its woody stems offered attachment points for climbing plants and shelter for small animals. When the plants died and fell, their decomposing tissue fed fungi and bacteria that supported the entire detritivore food web. One of the most interesting aspects of Neuropteris paleobiology comes from studying leaf damage patterns. Fossil leaves frequently show evidence of insect herbivory, holes, edge feeding, mining galleries, and galling. The diversity of damage types suggests multiple insect species specialized in feeding on Neuropteris, each with different feeding strategies. Some insects chewed holes through the leaf blade. Others mined between the upper and lower leaf surfaces, creating serpentine trails. Still others induced galls, causing the plant to produce abnormal growth that provided both food and protection. The extinction of Neuropteris followed the broader pattern of seed fern decline during the Permian. As climates dried and conifers evolved more efficient adaptations for water conservation, seed ferns lost their competitive advantage. Neuropteris persisted longer than many of its relatives, with some species surviving into the early Permian. But eventually, the changing world left Neuropteris behind. The delicate, water-dependent leaves that had been so successful in carboniferous swamps became a liability in Permian drylands. The lineage faded, and by the middle Permian, Neuropteris was extinct. The Ceronius tree ferns. Long before palm trees, before bamboo, and before any modern tropical plant, there was Ceronius, a tree fern that grew 10 metres tall with a trunk made entirely of roots. This wasn't wood, wasn't bark, and wasn't any structure that modern trees use. The entire trunk was a dense mass of tangled adventitious roots that emerged from a central core and fused together into a rigid column. From a distance, Saroneous forests looked almost normal, with straight trunks and leafy crowns. Up close, the anatomy was biological chaos held together by sheer mass. The root trunk of Saroneous was unlike anything evolution had tried before or since. At the centre of the trunk was a relatively small stem that contained the vascular tissue and produced the fronds. But this central stem was weak and couldn't support the weight of the large fronds that grew from its top. To compensate, Saroneous evolved a strategy of producing roots continuously from the base of the stem. These roots grew downward, pressing against each other and forming a thick, rigid mantle around the central stem. The outer roots hardened and died, creating a protective layer, while inner roots remained living and functional. The result was a trunk that grew thicker over time as new root layers added to the outside. The fronds of Saroneous were massive, some reaching 4 meters in length. They were typical fern fronds, compound leaves that divided and subdivided into hundreds of small pinnae. These fronds grew in a crown at the top of the trunk, creating a canopy that shaded the forest floor. Unlike many other carboniferous plants, Saroneous fronds remained green year-round in tropical environments, providing constant photosynthesis. Fossil evidence shows that in seasonal climates, some Saroneous species shed their fronds during unfavourable periods and regrew them when conditions improved. Reproduction in Saroneous followed the typical fern pattern, spores produced on the undersides of specialised fronds. These sporangia were clustered in groups called sori, which released millions of spores when mature. The spores were tiny and wind-dispersed, capable of travelling significant distances. But fern reproduction has a vulnerability. It requires water for fertilisation. The spores germinate into tiny gametophytes that produce sperm and eggs, and the sperm must swim through a film of water to reach and fertilise the eggs. This water dependency confined Saroneous to wet environments where moisture was reliably available. The ecology of Saroneous forests was unique. The root trunks created habitat structures that no other plant provided. Small animals could nest in the gaps between roots. Climbing insects and arthropods used the rough surface of the root mass as highways through the forest. When old root layers died and decayed, they provided food for decomposers while the tree was still alive. Fossil sites preserve evidence of insect burrows, fungal growths, and other organisms that colonized living Ciceroneous trunks. One of the strangest discoveries about Ciceroneous came from studying growth patterns in fossilized trunks. The root trunk didn't grow uniformly. Some sides added roots faster than others, causing the trunk to lean or curve. Over time, Saroneous could adjust its growth to compensate, straightening itself by producing more roots on the leaning side. This ability to self-correct gave Saroneous surprising stability despite its unconventional trunk structure. Fossil forests show Saroneous trees growing on unstable swamp substrates where conventional trees would have toppled. The geographic distribution of Saroneous was wide, with fossils found in North America, Europe, and Asia. Different species adapted to different environments. Some grew in permanently waterlogged swamps, while others colonized seasonal wetlands that dried out periodically. This adaptability allowed Saroneous to thrive throughout the Carboniferous and into the Permian. But like other Carboniferous plants, Saroneous couldn't survive the drying climates of the late Permian. The water-dependent reproduction and preference for wet habitats became fatal liabilities. By the end of the Permian, Saroneous was extinct. The legacy of Saroneous lives on in a strange way. Modern tree ferns use a similar strategy of root trunk construction, though on a much smaller scale. Tree ferns today rarely exceed 10 meters and grow primarily in tropical cloud forests where constant moisture supports their reproduction. They're living echoes of the Saroneous forests that once covered vast areas of the Carboniferous world, reminders that evolution sometimes revisits old solutions when conditions are right. The Williamsonia pseudoflowers. Evolution experimented with flowers long before flowering plants existed, and Williamsonia was one of the most elaborate experiments. These weren't true flowering plants, they belonged to an extinct group called Bennetites, but their reproductive structures looked strikingly flower-like. Williamsonia grew as small trees or shrubs with tough palm-like leaves and flowers that could reach 20 centimetres in diameter. These structures had colourful bracts, organised reproductive parts, and symmetry that mimicked true flowers so closely that 19th century paleobotanists initially classified Williamsonia as an early angiosperm. The plant itself was modest in size, usually growing between 2 and 4 metres tall, with a thick, stocky trunk covered in leaf scars. The leaves were long, narrow and leathery, adapted for conserving water in environments where seasonal drought was common. Williamsonia grew during the Jurassic and early Cretaceous, a time when conifers dominated forests, but flowering plants hadn't yet appeared. In this world of green and brown vegetation, Williamsonia's bright pseudoflowers stood out. The flowers were the plant's most remarkable feature. At the centre was a cone-like structure containing ovules, surrounded by a ring of pollen-producing organs. Enclosing both were large, colourful bracts, modified leaves that looked like petals. Some fossil specimens preserve enough detail to suggest the bracts may have been red, orange or yellow, though colour reconstruction from fossils is always speculative. The arrangement was bisexual, with both male and female parts in the same structure, exactly like many modern flowers. What makes this even more fascinating is that Williamsonia's flower-like structures evolved completely independently from true flowers. Bennetites and angiosperms shared a distant ancestor, but their reproductive structures developed through separate evolutionary paths, a textbook case of convergent evolution. The similarity suggests that the flower form has inherent advantages, perhaps for attracting pollinators or protecting developing seeds, that evolution discovered multiple times. Evidence for insect pollination in Williamsonia comes from several sources. The pseudoflowers were too complex for wind pollination, which typically favours simple structures. The colourful bracts suggest visual attraction, and the arrangement of reproductive parts would have made sense for an insect visitor that needed to contact both pollen and ovules. Fossil sites from the Jurassic preserve early beetles, flies and other insects that could have served as pollinators. Some researchers speculate that the relationship between Williamsonia and insects may have driven the evolution of more sophisticated pollination strategies that flowering plants later perfected. The seeds of Williamsonia developed within the central cone, protected by the surrounding bracts. When mature, the entire structure may have dropped from the plant intact, or the bracts may have opened to release individual seeds. Fossil seeds are rare, making the dispersal method uncertain, but the large size of the pseudoflowers suggests the plant invested significant energy in each reproductive structure, implying that seed production was limited, but each seed was well provisioned for survival. Williamsonia grew in diverse environments, upland forests, coastal plains, and even semi-arid regions. Different species showed different adaptations, with some developing thicker leaves for drought tolerance, while others had broader leaves for shaded understory conditions. This ecological flexibility allowed Bennetites to persist for over 100 million years, surviving multiple extinction events that wiped out other plant groups. But when true flowering plants evolved in the early Cretaceous, Bennetites faced competition from organisms that did flowers better. Angiosperms evolved faster growth, more efficient reproduction, and better relationships with pollinators. Within 20 million years, Bennetites went from dominant mid-canopy plants to marginal species clinging to unfavourable habitats. By the end of the Cretaceous, Williamsonia and its relatives were extinct, outcompeted by the very floral strategy they had pioneered. the lepidodendron spore swarms. The reproductive events of lepidodendron forests created one of the most dramatic natural phenomena in Earth's history. Spore swarms so massive they darkened the sky and covered the landscape in a yellow-brown haze. When millions of lepidodendron trees released their spores simultaneously, the air became thick with reproductive cells. These weren't gentle dustings of pollen like modern pine forests. These were biological blizzards that could blanket hundreds of square kilometres, creating conditions so hazardous that animals had to seek shelter or risk suffocation. The trigger for mass spore release isn't fully understood, but evidence suggests it was synchronised across entire regions. Climate factors, perhaps seasonal flooding or specific temperature changes, caused lepidodendron cones to mature simultaneously. When conditions were right, the cones opened and released their contents all at once. Each cone contained millions of spores, and each tree produced dozens of cones. A single lepidodendron forest could release trillions of spores in a matter of days. The spores themselves were tiny, measuring only a few micrometers across, but their numbers compensated for their size. Fossil sites preserve spore layers. Thin strata in the rock composed almost entirely of lepidodendron spores. These layers can be traced across hundreds of kilometers, showing that single reproductive events affected enormous areas. The thickness of these layers suggests spore deposits could accumulate to depths of several centimeters, creating a carpet that covered the forest floor and choked waterways. The ecological impact of these spore swarms was significant. Small animals and insects likely faced respiratory problems during peak release periods. Waterways became clogged with floating spores, creating anoxic conditions that killed fish and other aquatic organisms. But the spores also provided resources. Fungi and bacteria fed on the nutrient-rich spore coatings. Insect larvae specialised in consuming spores, and these specialists likely experienced population booms following major reproductive events. For lepidodendron, the strategy was effective. With such massive spore production, even a tiny survival rate meant successful reproduction. Most spores died within days, killed by desiccation, predation, or unsuitable landing sites. But the few that landed in appropriate conditions, wet soil with adequate light, could germinate and begin the complex life cycle that would eventually produce a new tree. The waste was enormous, but in stable carboniferous swamps, the strategy worked for millions of years. The fossil record also preserves evidence of spore germination. Some fossils show tiny gametophytes, the intermediate life stage between spore and adult tree, preserved in exceptional detail. These microscopic structures reveal that lepidodendron gametophytes were simple, consisting of just a few cells. They lived in wet soil, produced sperm and eggs, and after fertilization, developed into the familiar tree form. This life cycle, alternating between spore-producing trees and tiny gametophytes, is the same pattern modern ferns follow, though at a vastly different scale. Modern atmospheric modelling has attempted to recreate the conditions during lepidodendron spore releases. The models suggest that in the high oxygen carboniferous atmosphere, spore clouds could have posed fire risks. The spores contained oils and proteins that were flammable and, if ignited by lightning or forest fire, could have created explosions or rapidly spreading flames. Evidence for this comes from charcoal deposits that contain intact spores, evidence that fires burn through spore-rich layers. The end of lepidodendron's dominance meant the end of these dramatic reproductive events. When the climate dried and swamps disappeared, the synchronized spore releases became less effective. Without reliable moisture, germination rates dropped. Without widespread suitable habitat, the few surviving spores couldn't establish new forests. The biological blizzards that had been annual events for millions of years simply stopped, leaving only thin layers of fossil spores as evidence they ever existed. The Sigillaria chemical warfare. Beneath the ordered exterior of Sigillaria forests lurked a chemical arsenal that made these trees some of the most toxic organisms in the Carboniferous. Modern analysis of fossilized Sigillaria tissue reveals concentrations of alkaloids, tannins, and other defensive compounds at levels that would be lethal to most modern herbivores. These weren't gentle deterrents, they were chemical weapons that made Sigillaria wood, bark and leaves extremely dangerous to eat. The defensive strategy was so effective that few carboniferous animals could safely feed on Sigillaria, giving these trees a competitive advantage that lasted for tens of millions of years. The production of defensive chemicals was metabolically expensive. Sigillaria had to invest significant resources into synthesizing compounds that provided no direct benefit to growth or reproduction. But the investment paid off by preventing herbivory. While other carboniferous plants suffered constant damage from insects and early herbivorous vertebrates, Sigillaria remained relatively untouched. Fossil leaves show minimal evidence of insect-feeding damage, and fossilized Sigillaria wood lacks the boring galleries that riddled other tree species. The specific compounds Sigillaria produced can be inferred from chemical signatures preserved in coal. Alkaloids, nitrogen-containing compounds that interfere with nervous system function, appear in high concentrations. Tannins, which bind to proteins and make digestion difficult, are also present. Some fossils show evidence of phenolic compounds similar to those modern plants use to deter fungi and bacteria. This chemical cocktail made Sigillaria tissue nearly inedible. But some organisms evolved countermeasures. Certain carboniferous insects developed detoxification mechanisms that allowed them to feed on Sigillaria despite its defenses. Fossil evidence shows that a few specialized insect species targeted Sigillaria exclusively, suggesting they had evolved specific adaptations for processing toxic compounds. This created an evolutionary arms race. Sigillaria evolved stronger toxins, insects evolved better detoxification, and the cycle continued. The toxicity may have extended to Sigillaria's ecological impact on surrounding plants. Allelopathy, the release of chemicals that inhibit competitors, is common in modern plants, and evidence suggests Sigillaria practiced it extensively. Soil beneath Sigillaria groves shows unusual chemical compositions that may have resulted from toxic compounds leaching from fallen leaves and decaying roots. These compounds could have prevented other plants from germinating, explaining why Sigillaria forests had such sparse understories. When Sigillaria forests died and collapsed into swamps, their toxic compounds didn't disappear. The chemicals persisted through the coalification process, making carboniferous coal more toxic than coal from later periods. This has practical implications. Coal formed from Sigillaria forests releases more harmful compounds when burned, creating environmental challenges for modern coal use. The defensive strategy Sigillaria evolved 300 million years ago still affects the world today. The evolution of herbivores capable of eating Sigillaria may have contributed to its decline. By the late Carboniferous, some early reptiles had developed robust digestive systems that could process toxic plants. Fossil sites show evidence of these herbivores feeding in Sigillaria forests, breaking the plant's chemical defenses. Once herbivory became possible, Sigillaria lost its major advantage. Combined with climate change and the rise of more efficient seed plants, the breach in chemical defenses may have hastened Sigillaria's extinction. The Archaeopteris soil revolution. When Archaeopteris evolved deep roots that could penetrate bedrock, it didn't just change forests, it changed the entire planet's surface. Before Archaeopteris, soil was thin, unstable, and nutrient-poor. Rock weathering was a slow, primarily chemical process that produced little soil depth. Archaeopteris roots acted like biological jackhammers, forcing their way into cracks and fractures, expanding them, and breaking solid rock into fragments. Over thousands of years, this mechanical weathering transformed barren rock surfaces into deep, fertile soils, capable of supporting complex plant communities. The process worked through a combination of physical force and chemical attack. As roots grew, they exerted pressure on surrounding rock, widening existing fractures. When roots died and decayed, organic acids from decomposition dissolved minerals, further weakening the rock. Successive generations of roots penetrated deeper, each generation building on the work of the previous one. Fossil sites preserved root casts, impressions of Archaeopteryx roots in ancient bedrock, that show penetration depths of over 3 meters. This soil building process had cascading effects on the carbon cycle. Deep roots meant more rock surface area was exposed to weathering. Weathering reactions consume carbon dioxide from the atmosphere, converting it into dissolved minerals that wash into the ocean. The more soil Archaeopteryx created, the more weathering occurred, and the more CO2 was removed from the atmosphere. Some climate models suggest Archaeopteryx forests pulled so much carbon dioxide from the air that they triggered the late Devonian ice ages, cooling the planet by several degrees. The creation of deep soil also changed water cycles. Before Archaeopteryx, rain fell on thin soil or bare rock and ran off quickly, creating flash floods and leaving landscapes dry between storms. Deep soil acted like a sponge, absorbing rainfall and releasing it slowly. This stabilized stream flow, created permanent springs, and maintained wet conditions that allowed other plants to establish. Archaeopteryx didn't just survive in an environment, it created the environment that made its own success possible. The fossil record shows that soil depth increased dramatically during the late Devonian, coinciding with Archaeopteryx expansion. Early Devonian fossil sites show plant remains in thin soil layers rarely exceeding a few centimetres. Late Devonian sites show soil profiles several metres deep with distinct layers, topsoil, subsoil and weathered rock, similar to modern forest soils. This transition represents one of the fastest and most significant changes in Earth's surface environment in geological history. The effects on terrestrial animal evolution were equally profound. Deeper soil meant more habitat for burrowing organisms. Increased soil moisture supported more diverse plant communities, which in turn supported more complex food webs. Arthropods diversified rapidly in late Devonian soils, evolving new forms adapted for burrowing, feeding on roots, or hunting other soil organisms. The first soil ecosystems developed in the soils Archaeopteryx created. When Archaeopteryx went extinct, its soil-building legacy remained. The deep soils it created persisted long after the trees themselves disappeared, providing substrate for the next generation of plants. Seed plants and early conifers inherited landscapes fundamentally transformed by Archaeopteryx roots. Without Archaeopteryx's soil revolution, the forests that came after, including the great Carboniferous coal swamps, might never have developed. The Calamites' fire adaptation. In the high oxygen Carboniferous atmosphere, forests burned with an intensity unmatched in any other period of Earth's history. Lightning strikes could ignite fires that burned through entire landscapes, consuming everything in their path. Most plants had no defense against such infernos, but Calamites not only survived fire, it evolved to depend on it. Fire became part of its life cycle, an event that cleared away competitors and created perfect conditions for Calamites' regeneration. The plants turned catastrophe into advantage through a suite of fire adaptations that allowed them to thrive in an environment where other plants could only burn. The key was the rhizome system. While above-ground portions of Calamites burned completely, the underground stems remained protected. Buried beneath wet swamp mud, rhizomes survived even intense fires that sterilized the surface. Within weeks of a fire, new shoots would emerge from the undamaged rhizomes, carpeting burned areas with fresh growth. This regrowth was often explosive. Without competition from other plants, Calamite shoots could grow several meters in a single season. Fossil charcoal deposits preserve evidence of this fire regime, Layers of calamites charcoal alternate with layers of living plant material, showing repeated cycles of burning and regrowth. Some fossil sites show evidence of fires occurring every few decades, maintaining calamites' dominance by preventing other plants from establishing. This fire frequency was far higher than in modern forests, but the high oxygen atmosphere made ignition easier and fires more intense. The hollow trunk structure of calamites may have been another fire adaptation. When fires burned through calamites' groves, the hollow trunks acted like chimneys, channeling flames upward and away from the rhizomes. This protected the most vital parts of the plant while allowing the above-ground portions to burn away cleanly. After fires, the landscape was covered in hollow calamite stems standing like charred pipes, dead, but still structurally intact for months or years. Evidence suggests calamites may have promoted fire through its own biology. The plant produced substantial amounts of dry, flammable tissue. Dead calamite segments accumulated on the forest floor, creating fuel loads that made fires more likely and more intense. By increasing fire frequency, calamites created conditions that favoured its own regeneration, while preventing fire-sensitive competitors from surviving. It was ecosystem engineering through fire promotion. Animal communities in calamites' forests had to adapt to frequent fires. Fossil sites show that mobile animals, early tetrapods, large arthropods, probably fled ahead of fires or sheltered in waterways. Slower organisms, and those that couldn't escape, probably suffered mass mortality during fire events. But the post-fire environment offered opportunities. Fresh calamite shoots provided abundant food, and the absence of predators during the immediate post-fire period created safe feeding conditions. The end of calamites' dominance coincided with declining atmospheric oxygen levels and changing fire regimes. As oxygen dropped from 35% to closer to modern levels, fires became less frequent and less intense. Calamites lost its fire advantage. Plants that invested in fire resistance rather than fire dependence began to outcompete it. By the Permian, calamites was confined to marginal wetlands where fires still occurred occasionally, and even there it was being displaced by other plants. The chordate's pollen flight. Chordates revolutionized plant reproduction by evolving some of the most sophisticated pollen delivery systems of the Paleozoic. Unlike spores, which were tiny and depended entirely on wind for dispersal, chordate's pollen was relatively large, equipped with air sacs that helped it float, and produced in such quantities that fossil sites preserve pollen layers thick enough to see with the naked eye. This pollen didn't just scatter randomly, it travelled in organised patterns following wind currents, sometimes covering hundreds of kilometres before landing on receptive female cones. The pollen grains themselves were engineering marvels. Each grain had two large air sacs attached to the sides, creating a structure that resembled a tiny blimp. These sacs reduced the grain's density, allowing it to float longer and travel farther than simple spherical pollen could. Fossil pollen shows remarkable preservation of these structures, with the air sacs still visible under magnification. The design was so effective that conifers, descendants of chordates, still use the same basic pattern today. Production volumes were staggering. Male chordates cones produced so much pollen that when released, the air in chordates forests would have been thick with yellow clouds. Modern wind-pollinated plants like pines produce similar clouds, but chordates operate at a larger scale. Estimates suggest a single large chordates tree could produce billions of pollen grains in one season. Most of these grains never reached their target. They landed on rocks, water, soil, or other plants. But the wasteful strategy worked because sheer numbers guaranteed some grains would find female cones. The female cones were designed to catch airborne pollen efficiently. They produced sticky droplets at the opening of each ovule, creating pollen traps that captured grains from the air. When pollen landed in these droplets, the droplet was retracted, pulling the pollen grain into direct contact with the ovule. This active capture mechanism was far more efficient than relying on pollen accidentally entering the cone. It represented a major step toward the sophisticated pollination system flowering plants would later evolve. Fossil evidence shows that cordite pollen travelled remarkable distances. Pollen grains have been found in marine sediments hundreds of kilometres from the nearest chordates forest, carried there by winds that swept pollen out to sea. This long-distance dispersal allowed chordates to colonise isolated areas, establishing populations far from parent trees. It also meant chordates forests in different regions could exchange genes, maintaining genetic diversity across wide geographic ranges. The timing of pollen release was probably seasonal, triggered by specific environmental cues. In seasonal climates, chordates likely release pollen during specific months when winds were favourable and female cones were receptive. In tropical regions with less seasonal variation, pollen release may have been more sporadic, following rainfall patterns or other local climate factors. The lack of clear seasonal markers in equatorial chordates fossils suggests these trees had less synchronised reproduction than their temperate relatives. The success of chordates pollen systems influenced later plant evolution. When conifers evolved from chordates ancestors, they inherited and refined the air sac pollen design. When flowering plants evolved different pollination strategies, they still had to compete with the efficient wind pollination system chordates had pioneered. The innovation chordates developed, active pollen capture, structured pollen grains, massive production volumes, set standards that later plants had to match or exceed. The Glossopteris Gondwana dominance. The rise of Glossopteris to complete dominance across Gondwana represents one of the most extreme cases of botanical monopoly in Earth's history. Within a relatively short time period, geologically speaking, Glossopteris spread from isolated populations to continental-scale dominance, eliminating nearly all competing tree species. The fossil record shows this transition clearly. Early Permian sites preserve diverse plant communities with multiple tree genera, while late Permian sites contain almost exclusively Glossopteris. Something about this plant gave it an overwhelming competitive advantage. The key may have been cold tolerance. Gondwana during the Permian included large areas in polar and subpolar latitudes. Winter darkness lasted months, temperatures dropped below freezing, and permafrost locked soil water into ice. Most Permian plants couldn't handle these conditions. They either required year-round growing seasons or couldn't survive freeze-thaw cycles. Glossopteris evolved specific adaptations for polar life, deciduous habits to survive dark winters, cold-resistant roots, and biochemical protection against freeze damage. This cold tolerance allowed Glossopteris to colonize areas other plants couldn't reach. Once established in polar regions, Glossopteris spread toward the equator, moving into temperate and eventually subtropical zones. In each new environment, it encountered local plant communities that had evolved for warmer conditions. Glossopteris outcompeted them through superior growth rates, more efficient seed production, and better stress tolerance. Within a few million years, Glossopteris had displaced most rivals across the entire supercontinent. The ecological impacts were severe. By monopolizing forest canopies, Glossopteris controlled light availability, soil composition, and water cycling across Gondwana. Understory plants had to adapt to Glossopteris filtered light or go extinct. Herbivores had to either learn to eat Glossopteris or starve. Decomposers had to process Glossopteris leaf litter or find other food sources. Entire ecosystems reorganized around this single plant genus, but monopoly created vulnerability. When the end Permian extinction struck, Glossopteris had no backup species, no similar plants that could fill its niche if it failed. When volcanic eruptions, climate change and ocean acidification pushed conditions beyond Glossopteris tolerance, the entire forest biome collapsed. There were no alternative forest species ready to take over. Glossopteris had eliminated them all. The result was ecological catastrophe across Gondwana, with forest ecosystems disappearing completely in some regions. The recovery took millions of years. Triassic plant communities that eventually refilled Gondwana forests bore no resemblance to Glossopteris ecosystems. They were dominated by entirely new groups, seed ferns, early conifers, and eventually cycads, that had evolved during or after the extinction event. Glossopteris left no descendants and no ecological legacy except a lesson about the dangers of monoculture. Dominance and diversity are both survival strategies, and Glossopteris chose dominance. When conditions changed, that choice proved fatal. The arborescent Lycopsid collapse. The fall of the giant Lycopsiads, Lepidodendron, Sigillaria and their towering relatives wasn't a gradual fade into obscurity, but a catastrophic ecosystem collapse that reshaped the planet's surface. For 50 million years, these scale trees had dominated wetland forests across every continent, their shallow root systems stabilising vast swamps and their massive trunks storing carbon that would eventually become the coal deposits we mine today. Then, within a geological instant of just a few million years, they vanished almost completely. What remained were their tiny descendants, club mosses barely reaching ankle height, living proof that evolution sometimes takes giant steps backward. The trigger was climate change, but not the kind we typically imagine. During the late Carboniferous, around 300 million years ago, the continents were colliding to form the supercontinent Pangaea. This massive tectonic reorganisation altered ocean currents, changed precipitation patterns, and shifted climate zones across the globe. The warm, wet conditions that had sustained Lycopsiad swamps began to break down. Rainfall became seasonal rather than constant. Water tables dropped. Vast wetlands that had existed for millions of years began to drain. Lepidodendron and Sigillaria were catastrophically unprepared for these changes. Their shallow stigmaria root systems, which had been perfectly adapted for waterlogged soils, couldn't reach deeper groundwater. When swamps dried even temporarily, the roots died of desiccation. The tree's reproductive strategy, releasing massive quantities of spores that required constant moisture to germinate, failed in seasonal environments where dry periods killed young gametophytes before they could complete their life cycle. Even their structural design worked against them. The soft, spongy interior of Lycopside trunks needed constant water pressure to maintain rigidity. When water became scarce, trunks collapsed under their own weight. But the collapse created opportunities. As Lycopsid forests died, they left behind landscapes covered in dead wood and empty ecological niches. Seed plants, conifers, seed ferns, and early cycads had been waiting in the wings, confined to drier uplands where Lycopsides couldn't compete. These plants had evolved adaptations the Lycopsides lacked: deep root systems that could reach groundwater, seeds that didn't require constant moisture for germination, and wood structures that remained rigid even during droughts. When the swamps dried, seed plants moved in. One of the strangest aspects of the collapse was its effect on atmospheric chemistry. Living Lycopsid forests had been massive carbon sinks, pulling CO2 from the atmosphere through photosynthesis and locking it away in their tissues. When they died, that carbon didn't return to the atmosphere immediately. It was buried in swamps and eventually became coal. But the new seed plant forests that replaced Lycopsids were less efficient at carbon storage. They grew on better drained soils where decomposition happened faster, returning carbon to the atmosphere more quickly. The net result was a shift in the carbon cycle that may have contributed to the warming trend that marked the transition from Carboniferous to Permian. The surviving Lycopsids tell a remarkable story about evolutionary constraints. Modern club mosses, descendants of the giant Lycopsids, remain committed to the same basic body plan and reproductive strategy that doomed their ancestors. They still produce spores that require moisture, still have relatively shallow roots, and still prefer wet environments. Evolution didn't fix the vulnerabilities that caused the Carboniferous collapse. It simply scaled everything down. Small size became a survival strategy. A plant a few centimetres tall needs far less water than an 8-metre giant, can complete its life cycle in a single season rather than decades, and can persist in microhabitats too small for trees. The most profound lesson from the Lycopside collapse is about the dangers of over-specialisation. For 50 million years, Lepidodendron and Sigillaria had perfected an adaptation for one specific environment. They became so efficient at exploiting waterlogged swamps that they outcompeted everything else in those habitats. But that narrow specialisation made them vulnerable. When their perfect environment disappeared, they had no flexibility, no backup adaptations, and no time to evolve new strategies. The giants that had dominated continents went extinct, while their less specialised relatives, smaller, more flexible, more adaptable, survived by accepting mediocrity over mastery. The coal deposits left behind by the Lycopsid collapse are humanity's inheritance from this ancient catastrophe. Every piece of coal burned in power plants worldwide represents the compressed remains of these fallen giants. The energy that powers cities, factories and homes comes from forests that died 300 million years ago, when the climate changed and evolution moved on. The Lycopsids couldn't adapt quickly enough to survive, but their fossilised bodies still fuel the modern world. In a strange twist, the plants that failed to survive climate change are now contributing to our current climate crisis, a connection that spans 300 million years and links the Carboniferous collapse to contemporary environmental challenges.